The method can answer key questions in mitochondrial function field, such as ATP level and oxygen consumption rate. The main advantage of this technique is that it's easy to perform and it allows for special evaluation of cellular mitochondrial function.
To start this experiment, seed 20,000 HepG2 cells in a glass-bottom petri dish. Add beta-hexachlorocyclohexane, or BHCH, and incubate at 37 degrees Celsius and 5% CO2 for 24 hours. Prepare 1 millimolar mitochondrial green fluorescence probe solution by adding anhydrous DMSO to DMEM cell culture medium. Then add 1 milliliter per dish of this solution to the cells in glass-bottom petri dishes and incubate at 37 degrees Celsius for 45 minutes. After incubation, remove the mitochondrial green fluorescent probe solution from the cells. Add 1 milliliter per dish of freshly prepared 37 degrees Celsius DMEM cell culture medium. To detect green fluorescence in mitochondria, observe the cells under a confocal microscope. To evaluate levels of cellular ATP, seed 20,000 HepG2 cells in 6 well plates. Add beta-HCH and incubate at 37 degrees.